So the next session is the panel discussion. So we've been running this for a couple of years, and the hope here is for this to be very interactive. So please do remember to submit your questions through the Q&A button at the bottom of Zoom. The title is Standardization in the Breath Research Field. How as a community can we benefit from each other's data? Again, I think it's really important that we work together as a community to help bottom out issues around things like standardization and what some of the best practice are. So we're welcoming back Morad and Orna from this morning's session, and also delighted to welcome Paul Thomas, who's Professor of Analytical Sciences at Loughborough University. His general interest in determination of volatile organic compounds and a particular interest in volatile breath biochemistry, as well as clinical studies with respiratory and oncology teams. Paul's been looking at breath and volatile organic compound applications in emergency rooms and austere settings. The resultant multidisciplinary studies have integrated arm mobility techniques into remote piloted airborne systems, skin analysis for contamination, and rapid breath testing for mass casualty applications. So Paul, we can see you, and we also have Joachim Plau, who's retired from the US Environmental Protection Agency after 32 years as a research scientist focusing on human systems biology of exposure. He's adjunct professor at the University of North Carolina School of Public Health, currently serves as the editor-in-chief of the Institute of Physics Journal of Breath Research, consults with US NASA projects on jet fighter pilot breathing assessment and International Space Station air quality, and he's a founding member of the International Association of Breath Research. So again, the hope here is that we'll get a lot of questions coming through related to the topic. We have a few, and I want just to kick off with the first question to stimulate the discussion and the debate. And as an expert in your field, in your view, how many VOCs are on breath? And how confident are you that these originate on breath and are not from other sources? I often read papers that say there's hundreds of thousands of VOCs on breath. And the question is, how do we know and what's the proof? Maybe we can start with Paul on that one, please.
This is a little bit like analytical limbo, isn't it? I mean, just how good is your kit is the answer to that question, and what's your sample volume and the power of your pre-concentration. I think the ubiquitous panel of breath volatiles probably isn't that large, and it would be good if we could agree what that is. So something that you will find in every person's breath sample, I think that's a manageable number. I'm going to say less than 1,000 at this stage. That we can detect in the near and medium term. But then on top of that, you've got huge variability in what's going to be present in your breath, depending on your exposome and everything else that's going on in your life. And anyone, I think, who's done any breath study is going to have lots and lots of information that they junk at the outset of the data modeling because it's rare data. That doesn't mean to say it's uninteresting data, it's just that we don't have the tools to process it yet. So it's the rare compounds that I think the discoveries are to be made in the future. And how many of those there are, I'm not prepared to have a guess at. I think if you think about how you'll generate a catabolite from an exposure, and then that will be absorbed into your gut, and then your gut metabolism will process that and put it back into your body, and then you'll have secondary processes. Who knows just how that works on, and then your enzymes and oxidation processes get to act on that. So the actual number of rare compounds, I think, is likely to be very large indeed, and keep people busy for a long time yet.
Excellent. York, I was wondering if you could pick that up.
Yeah, so yeah, Paul's right. I mean, you can find anything you want if you look hard enough. One thing I want to point out, and this is something I always try to reinforce, is all breath analysis is stratified, which means that you never get everything in a single analysis. It all depends on how you look. You can, if you're doing VOCs, you may stratify nonpolar versus polar, depending on your mechanism. You may stratify higher rather than lower compounds. And so this idea that there's a kind of a magic black box that tells you what every VOC in breath is, I think is wishful thinking. And I would love to see that. But, if you use a sorbent material versus a sumacand versus some kind of an adsorptive surface, you're going to stratify your sample before you even can make any kind of analytical decisions, okay? A lot of us have recently going towards the time of flight or some of the Orbitrap methodologies, and primarily starting with some kind of a liquid sample, say condensate or whatever, completely different stratification than VOC. So I want everyone to kind of keep that in their mind, that you can very easily miss a super probative compound because you don't have the capability to look for it. And this becomes a real problem when you're doing non-targeted or discovery analysis. At EPA, before I retired, we like to believe we could find everything by LC Orbi or LC QTOF, and we could find everything that we could have sampled in the first place, right? And so this is just the cautionary tale. There's no such thing as the Star Trek tricorder, as Wolfie Mikush always used to say. And so, and I have a few other things I might talk about later, but for the time being, as far as how many compounds there are, I recall one of the posters was doing GC-GC. Well, this opened up a huge can of worms for us, right? We go in linear chromatography into GC-GC. We're going, there's like 10,000 more compounds in here than I thought, So it's hard to hang a number on this. It's lots, and the harder you look, the more you find.
Yeah, brilliant. Orna, do you want to pick up anything additionally to add there?
Yes, actually, it's a very painful question, and I agree with what been said by Joachim and Paul, that the more you look for, the more you will find. But I feel like maybe the bigger question, or the more painful question, is the variability of these VOCs. Like, you can find 10 of thousands of VOCs, but how significant are they? How can we determine their significance and their source? I think the variability and their distribution is something very crucial we need to look deeply into. It's also very important if we're trying to look for differences between healthy and sickness, in terms of if the difference is hidden inside the distribution, then it's almost impossible to look for it. So it's not only the number of VOCs, but also their distribution in healthy and in sickness. A very painful and important parameter, I think.
Thank you. Morad?
Yes, it's a very interesting question, and I think it's somewhat theoretical and a bit provocative. But I think it raises a very important question, because I started in the field about 12 years ago. I'm not as experienced as Paul or Yok. Orna and I started more or less at the same time. And if you monitor the literature, you could actually see the change over time about the number of VOCs that people detected based on, like been mentioned, if it's tubes, reusable tubes, are they conditioned, is it in bags, and which analytical instrument you use. And I think that drew a very unrealistic picture, because if you read most of the reviews, you'll start typically the first sentence starts with exhaled breath contains over 3,500 VOCs. And I think this is what the researcher actually would expect to see in their samples. But when we continue and we start better understanding the background and the sources of those compounds, we actually learned that no, it's not 3,500 that we expect to see in a breath sample, despite, and I agree with Paul on his comment that xenobiotics would differ between different nations, races, countries based on diet and exposure and so on. But I don't think that this variability ranges in the thousands of volatile organic compounds that we might end up finding on breath. But I think to release some of this pressure, and I think to make a better clarity when we publish our data in the future, instead of mentioning the number of peaks on the chromatograph, which is typically thousands, we could focus on the ones that have substantial signal to background that minimizes the risk that it's a contaminant or just a peak from the tube or the bag or the ambient. So by filtering the data based on understanding and testing against the background, we could, and we've seen this today in the presentation where we looked into over 2,000 features, and 500 of those we had very high confidence they're actually coming from breath. And that makes great difference going forward in terms of standardization in the field, I believe.
So that kind of leads me on to a second question, and Paul touched on it a little bit already. So the comment that just because something is rare doesn't mean it's uninteresting. But I think that the thing that seems obviously true is for it to be interesting in some regard, it does need to be a compound that's actually on breath. So that could be xenobiotic, an exogenous compound, an endogenous compound. And we know quite often that there is a lot of background contamination. So, to me, it does seem logical that we need to be focusing on compounds which are truly on breath, irrespective of the source of those, and narrowing down the set to do studies on that. And one of the observations that I would make is that quite often a particular sampling approach or analytical technique will be used in a clinical study. But actually, what doesn't tend to get reported for that combination, that end-to-end workflow is, how many compounds are actually being detected on breath in very simple healthy volunteer populations prior to launching into running a clinical study. So I'd be interested in the perspective, and it was a question I asked in the session this morning, of whether or not we do need to start to report in a fairly standardized way what the basic performance is of some of these end-to-end systems and workflows, looking at what is actually on breath compared to things which are analytical artifacts in the background. So I might start with Jock on this one, please.
So one of the things that we've struggled with is exactly what you're talking about, is like, what is breath and what is something you were exposed to 6 weeks ago? What is something that happens to be lurking in your machine? What is, finding the things that are lurking in your machine are pretty straightforward. You just introduce a clinical air standard with 5% CO2 and water vapor, and you see what kind of junk comes out. That doesn't really address the carryover issue, but so let's leave that off the table for now. I completely agree that humans breathe out a lot of stuff that their metabolism does not make. That does not, however, at least in my opinion, mean that these compounds are not useful. My little example is we did a little study with the Italians on people who underwent surgery and had sevoflurane as the anesthetic. And sevoflurane is completely exogenous, but its level post-operative and its primary metabolite, hexafluoroisopropanol, tells you what the liver is doing, right? You yourself will never make these fluorinated compounds. But this probe or this, in this case, kind of completely serendipitous probe, could show when the liver started kicking in and doing its job again. And it was really kind of fascinating because some of the patients that had liver shunts, and you could see in the profile of the metabolite when the liver started working again. And so, you know, all the folks that are measuring limonene, for example, you know, you say, well, limonene is an exogenous compound. It's, you know, you're just exposed to it. But how your body handles it might be the signal. Okay. So MTBE, the exogenous compound they put into gasoline for a while in the US, had a metabolite, tertiary butyl alcohol. And you could see it in blood, breath, and urine. It was a really cool combo, because a lot of times the metabolite only shows up in the urine, and right. But that was a really fascinating compound to use, not because you could ever make this stuff yourself, but because it was a probe into what else was going on in the body. So, you know, I always worry when people completely disregard the exogenous compounds when they pop up as a case control discriminator. The problem is, in one place it'll be that compound, in another place it'll be a different compound. You know, I don't know if you remember Mike Phillips's early articles where like CS2 and some of the freons were popping up as markers in case control, and people like me, I admit it, would write, well, this is ridiculous because, you know, everybody knows. And I've changed my mind. You know, I think Mike was pretty good back in those days. And he just didn't explain it well. Anyway, that's all I got on that.
Yeah, and I think it's interesting, and we came to a similar type of conclusion about, you know, the potential utility of exogenous compounds. You know, they can be used in diagnostic applications. I suppose the broader point, though, is that in order for it to have any potential utility, irrespective if it's endogenous or exogenous, do we agree it must be a compound that's actually on breath as opposed to in the background, which is often what we find within the chromatogram? So it's just to kind of refine the question a little bit. Morad, do you want to kind of pick it up?
Yes, I think it's a good point, and it's completely different. I'll speak for myself for how I used to treat data 10 or 12 years ago, where oxygenous compounds were the enemy. But now they were in the literature, in papers. We excluded this, we excluded that because it's exogenous, so it cannot reflect any metabolic pathways. And thus it cannot be any meaningful. But I think today, and Yok, you mentioned limonene. I think limonene is the most brilliant example of why we should look into exogenous and endogenous compounds equally. But as Billy said, if it's not coming from breath, without doubt, then it cannot reflect any biological activity or metabolic activity. And you said previously, junk that we might have been exposed to weeks ago. I mean, yes, it could be the case, but if this junk is being metabolized by enzymatic activity or being processed by the kidney or the liver, then just like the story you just told, we could learn about some endogenous activity. And that's by definition, this is what biomarker is. It's a characteristic that we measure objectively and indicates a biological or physiological activity. And therefore, I don't think that we need to distinguish between exogenous and endogenous. We need to more focus on, is it on breath, or it's actually coming from breath, or it's just a compound that is living on my tube, on my back in the ambient and just appearing on the chromatogram. And since most of the, I would say, promising data that has been published in the field, mainly focused in combination, linear combination and other combinations, machine learning, and so on, that even increases more the risk towards false discovery, because it's enough that one of the compounds that have been used in that model are not actually coming from breath, then this data will never be replicable or be validated going forward. And therefore, this distinction is, in my opinion, much more important than looking into endogenous or exogenous.
So, Paul, you used a lot of mobility as well as GC mass spec. So, it's the same question again. Do you think it's worth us as a field trying to look for whatever analytical platform, whatever sampling approach, reporting on the number of compounds that are truly on breath compared to blank prior to undertaking any clinical studies?
I think so. At the moment, we look at the cumulative frequency distribution of the signals we get from breath, and we know that a typical breath sample's got a fairly well-defined cumulative frequency distribution of the number of compounds, the distribution of intensities, where they arise in the retention index scales we're working with. And if it doesn't meet that specific threshold, it's a dodgy sample for a start. And we saw this in the peppermint study for sure. And we saw the effect of people putting HEPA biofilters in sample lines, and how the cumulative frequency distribution and the number of compounds just crashed the moment you start putting filters between the mouth and the sample medium. Bad thing to do, I think. So for sure, these basics, I would say, would be good to see them as supplementary at the very least. How you can prove the knife fight that sometimes it feels like with reviewers when they're trying to get you to prove things to the umpteenth and nth degree that what you're seeing is verifiable, and you're thinking, well, I only have a lifetime and maybe this amount of money. What is beyond reasonable doubt essentially is the challenge, and what's a reasonable qualification. But I think it's appropriate to talk about a threshold level. I'm hoping to publish some work soon about what happened when we looked at background levels over more than 2 years in a range of clinical settings. And how many compounds did we see, and what were the background concentrations we saw? And that there seems to be a sort of background low-level miasmic fog of VOCs present in every setting. And I can tell you now that you can see all the key biomarkers of disease in the background of a clinical room for sure. And if you're not careful in terms of ventilation rates of that room and times of day and room occupancy, lots of opportunity for misleading artifacts from the background in these things. But there is a sort of baseline threshold level, and above that level, you can be pretty confident that it's breath. And so you can say that if you're going to be working in this range of concentrations, there's a confidence, certainly as far as I'm concerned, that this isn't a background contaminant you're working with, because they tend to fall after more than 240 observations across all days of the week, all times of the working day, different people, different teams, below a certain threshold level, then that is likely to be coming from breath or coming from the instrument. And proper QC and proper analytical quality by design with regular QCs and blank checks will edit out those artifacts, or should do. But certainly the cumulative frequency diagram on what is the threshold you're working at, what are the concentration thresholds you're working at, I think.
So in terms of those thresholds, I don't know if you saw Murad's talk this morning, so we tend to think of 3 standard deviations above the background. I don't know if you have any kind of particular cutoffs or rules of thumb that you use in your own work to define what's a non-breath compound.
So what we found is that the background on the day can be quite variable, and to say what that background is problematic. So we tended to look at what's the average, what's the upper 75% quartile, or 1.96 level, and this was the sort of upper threshold where it was unlikely to ever see a background above this level. I don't want to give a number at this moment because I've not had my final reviewer comments back yet, whether they're actually going to allow me to make this claim. I keep my fingers crossed. It's been a while, but above this level, we just didn't see it. It just did not happen, regardless of when you sample. But if you look at the background level on the day, that was introducing a significant variability as to what constitutes on breath within your study. And now you've got an uncontrolled factor coming into your study that says, well, because we saw this patient on this day with a low background level, we're going to accept this as on breath, whereas in a fortnight's time, we may have rejected that. So that's why I'm saying threshold is a useful thing to look at this stage.
Excellent, thanks, Paul. Orna, should we be doing routine comparison and calculation of compounds on breath prior to running clinical studies?
Yes, definitely. I think we all here agree that the first thing is to make sure that we're testing breath VOCs that comes and originate from breath. Whether endogenous or exogenous, I agree with you. It doesn't really matter. Just we need to make sure it comes from breath, and we need to. I think we need to agree on the criteria of what are those breath VOCs, whether 3 times standard deviation, whether other threshold criteria, but we need to do it, and we need to validate this on a daily basis, because from our experience, we also see a big variability during the day. Even at the same location, during the day, you see variability. And more interesting is that some of the breath VOCs appear in the same, that we know comes from breath. We see them at the same level in the room also. And the reason for that is because people emit this to the room. So it comes from breath. It for sure comes from a human physiology and different, even known biochemical pathways, but you see this at the same level in your room. So you also need to make sure the room is well ventilated and things that you've mentioned before. So this calibration and tests need to be performed on a daily basis, for sure. And of course, we need to decide on the threshold, maybe even different threshold for different VOCs.
So I think there's a few interesting points that have been raised as well, and just reflecting back, in the yesterday within the conference, today within the conference. So, when we're trying to discover novel biomarkers and doing global analysis, you don't know a priori what you're necessarily going for. So that's where things like background contamination, controlling artifacts, background is very, very important. And I think that came up quite a lot yesterday within the conference, and this morning within the conference as well. We need to control for a lot of these factors, and that's ultimately one of the big reasons to try and drive standardization and reporting, et cetera, et cetera. I could characterize some of this afternoon's talks where people are deliberately using exogenous probes, introducing them into the body, targeting known pathways or biological processes or disease processes, and looking for completely unique compounds at very high fold change because the biology is understood there. So again, it's looking at those examples like limonene and D5-ethanol, et cetera, and saying you deliberately want to use those exogenous compounds. And again, if you look at the types of breath tests which are currently routinely used, H. pylori, some of the stable isotope tests, digestive health, by and large, some exogenous substrate has been added to the body. So my kind of provocative question is, should we bother at all with global analysis? Or should we just take a much more bottom-up hypothesis-driven approach of trying to understand the underpinning biology and hit it with specific exogenous substrates? So we'll start with Morad on that one.
Yes. Well, I have to say we need to stick to the global analysis, mainly because I need to maintain my income, Billy. Now, seriously to the question, I think the approach with the evokes is just genius. I think if we could target very specific biological or metabolic pathways with known compounds that we could administrate on high concentration, overcoming any sources of variability, background, and so on. I think it's a great and has been shown how successful this approach could be. However, we need to keep in mind, and if we go back to the limonene, limonene wasn't, I mean, the initial indication that limonene is or could be implicated with liver disease was through discovery. And I think, just like we said before, I think that the evoke approach did not grow rapidly before. It's mainly because exogenous compounds were looked at as the enemy. But today that we're looking at those, just like we're looking into indigenous compounds, I think we're realizing that those could be actual biomarkers. And I think by optimizing discovery platforms also for such range of compounds to monitor, understand the behavior, the variability of exogenous compounds in the population, I think it's a quick route to the identification of new probes, new ideas, new metabolic pathways that then could be targeted in vitro, in cell cultures, developing those probes specifically. But I think it's almost, I don't want to say impossible, but it's really difficult to predict upfront which exogenous compound could be linked to which metabolic pathway. But if you see it elevated or behaving differently in a population, you start by putting the hypothesis together and working towards understanding and developing such a test.
Yeah, and I think as well, leading on the Orna, if we approach it from the biology, then we do know things like oxidative stress lead to the production of aldehydes and those types of compounds as well. So maybe it's a combo of both. So Orna, do you want to kind of pick that up? The why we do global discovery versus just sticking probes in for diseases.
Yeah, I think that the evoke approach is brilliant. I think it's the future. Like if I think about breast analysis in the future, I think this is what's going to rule in this area. But I think it's right now it's still premature, and as Morad mentioned, probably the VOCs that we would like to use, that we will use as eProbes, will come from the untargeted analysis and the discovery analysis that we are now performing. It's not only knowing the mechanism that leads to production of VOCs, is also the knowledge of how to initiate such mechanisms in the body to produce the target VOCs that we want to see. It's complicated, though I think that if we will be able to tackle this, will probably be the future of breast analysis.
And Jock, it sounds with the example of sevoflurane. So that was a observation made from doing global analysis or target analysis, where you then try to figure out the biology as opposed to the other way around. So interested in your perspectives in this question.
Yeah, well, I have two things I want to say. First one is to address the exogenous versus endogenous. You've got to realize every endogenous compound is somebody else's exogenous compound, right? So there's a blur right there if you're in a crowded room.
Yeah.
So on the subject of discovery versus targeted analysis, we've been worrying, by we, I mean my old we, we've been worrying about this at EPA for about 12 years. And the classic example there was we were targeting perfluorinated compounds, PFAS and PFOA, in the environment. The company switched away from those, and yet we were continuing to target those compounds, going, hey, look how great this is, they're all going away. And then we realized by doing discovery analysis in the same samples using Orbitrap and QTOF, there's 100 new ones, okay? So all I want to say is there is huge value in what I call discovery analysis and what other people might call non-targeted. We got to keep doing that because new things happen. So I think that's really, really important to keep in mind. Now, with respect to, I guess you're calling EVOC, we used to call them probe molecules. I think that's a really clever way to go about it. And again, from the perspective of toxicity testing, this is something we developed at EPA. Instead of using people, we used cell lines. So we took, say, lung cell lines and transfected them so they could metabolize like a liver cell line. And we would put a probe molecule through there, monitor the metabolite, and then put the fairy dust that we were worried about in there to see if it disrupted the metabolic activity of the cell. I think that is valuable. But from the perspective of toxicity testing, which is not so much what we do other than as a breath approach, the mainstream breath is, can we figure out if somebody has a preclinical cancer or disease state, right? But again, one of the things we run into in our clinical studies is the number of subjects we have. Human subjects are expensive. They're hard to get. They're very complicated. And if we want to deduce metabolism, it had become my humble opinion to try to push things towards cell lines. The problem we have is you can't really use immortalized cell lines. You have to use wild type, because the immortalized ones, we've also changed the metabolism. So sometimes they don't behave the way you expect them to. But I think in the global sense of breath research, we should always consider, is there some way we can use cellular breath rather than systemic breath to kind of figure things out? Ultimately, of course, we have to use people breath. But people breath studies are expensive and hard to do, whereas cells, you don't need an IRB to kill off a few cells, right? So again, my thing is we cannot lose discovery analysis. I think when you get into outpatient clinical, yeah, you're going to be looking for compound A, B, and C. You're going to have a $100 detector, and you're going to monitor. It's like for diabetes, you monitor glucose. You're not going to monitor sucrose, and you're not going to monitor acetone. All you care about is that one thing. But for research, I think we have to keep our eyes open and our ears open and our minds open.
Yeah, that makes a lot of sense. Go back on the in vitro models we saw with some of the speakers earlier. Even with take a probe-based approaches, it's much more straightforward to get initial pilot data, administer probes into into simple in vitro models. And to your point of Ryan, making sure that we keep doing global analysis, non-targeted analysis, as you mentioned earlier, as analytical methods improve, as we have a range of different methods that give us broader coverage of breath as a sample matrix, have different sorbent materials or sampling methods. It means that we want to keep applying the best tools in those discovery settings. Paul, do you have an opinion on this? On should we keep plugging away or just go straight to the underpinning biology?
Both and everything. It depends what you're taking the breath sample for. I mean, you can see that you've got, in terms of utility, you have a screening breath test where do you have indicators, do you have presumptive markers of possible bad things going on in your life versus a functional breath test versus a differential diagnostic. They will all have different approaches and different best methods for implementing those. In terms of research and discovery, what is it our neurolinguistic program is? Tell us, if you do what you've always done, you'll get what you've always got. There's no way of advance once you decide we're going to limit ourselves by our limited understanding and the ability with which we can generate hypotheses to test. I'll give you an example for that. We tend to use siloxanes as a measure of the degradation of our system and or contamination, and sometimes they're helpful for attention indexing purposes. Until I came across a siloxane from a herbal oil and a herbal medicine in Sri Lanka that people were using to treat pesticide poisoning with. And then I'm thinking, well, that's a whole new basis of chemistry there. I've never seen it in a breath sample before. And how did it get there? And what does it tell us? And how might we exploit it? So there's always, for a long time yet, that opportunity for discovery and realization that moves the thing forward. I think I come back to, it depends what you're trying to accomplish here. And they all have their role to play. So I'm listening to what Jörg says here. And at this stage, I'm going to say there are some really important molecules that we're not seeing a lot of in breath yet. And I'd really like to see some putrescine and cadaverine and dimethylamine in breath samples, and for them to be reported really as a measure of infection and tissue damage. They are first-class markers. Our methods aren't picking them up properly at the moment. And we should be asking the question, what are we missing? What's on the menu that we know should be there that we don't see yet? And it's not until we've got the full palette, the full rainbow, if you like, of chemistries consistently present in our data, can we then start to pick and choose?
And that leads us into, there's an interesting question from Scott, and I think there's a bit of nuance to what he's asking. It relates back to the question of standardization. When there's so much development, should we consider standardizing? And I think, just picking up on what you're saying there, Paul, we want to keep pushing the envelope from the point of view of sample collection, analytical technique, because we want to have as broad of an aperture as possible to capture and see as many things as possible in breath. So I think here, when we talk about standardization, the question probably isn't around standardizing per se on a given analytical approach, a sampling approach, or anything like that. But reflecting back on Patrick's talk yesterday morning, when he, I think, was advocating standardization around things like reporting, for instance. So at the outset of a study, agreeing what the protocol is going to be. So trying to overcome, the problem that he was stating, that 96% of studies basically say it works, which can't be the case. That's rarely the case in science. So I think the question is more about, should we be aiming to try and standardize what we report? So, the reporting again of how many compounds may be on breath with a particular end-to-end workflow, making sure that we've defined the analysis at the outset, as opposed to just trying to find, what may be different in the, between the case control population. So I want to kind of loop it back to that and focus on not standardization of hardware, but should we standardize what we report and how we report? So maybe Orna, to kick us off on that one, please.
Yes. So I think it's crucial to standardize. I will focus on the reporting because this is what you were asking. I think also the collection should be to some extent standardized, though I appreciate that for some diseases you want the inner part of the breath, and for some you might want the upper respiratory breath. But I will put this aside and I will focus on reporting. And I think it's very, very important to have a standardized way of reporting, starting from the study design, the way the number of samples we include in each group, the groups, how we define a control group. Are those totally healthy control group? Are those people with similar conditions should be the control group? We need to standardize the statistical analysis we perform, even comparing the way we analyze data to the way Alston, for example, analyze data. There are differences. And I think it's very important to understand these differences and to find a way for us all to report the same way. It will make it, I think, more easier to replicate studies and to compare between studies. I find it now very, very difficult to compare and to conclude anything meaningful. When I look at many papers, I see that each paper refers differently to the data and analyze it differently and report it. Some report in PPB, some report just in trends, some report in abundance. It's really, really difficult to compare and to conclude from things from the literature, actually. Breath analysis is something that we're doing for, I don't know, 30 years, and still I feel like there's not enough consistency in the way results are presented, and it's a pity. I think we all lose from this. We can benefit much more if we all be unified in this perspective.
Thank you. Yoch, do you want to pick that up? So we maybe don't want to standardize hardware analytic methods, etc., but should we be thinking about standardized reporting?
As you can imagine, I've had like 40 years experience with standardization, setting regulations, turning off $1 billion of highway funds for Texas because they didn't do something, in the air. So this idea of standardization, I think, is more valuable when there is like regulation or something important, like you are going to have a biopsy, or right. I think for us, we have two levels, and I think Orna really put it well. We have the instrumentation and how we arrive at the number, and then we have the how do we look at that number. Okay, these are two different things. How we look at the numbers, case control, longitudinal, cross-sectional, whatever it is, I think we have a better chance of standardizing that. If many of my colleagues, we have like, I don't know, I used to touch base with a couple hundred laboratories across the country. Most analytical laboratories are not breath focused. In fact, hardly any, but they can do breath analysis. So if I say, hey guys, you're doing breath analysis, what do you got in your lab? And I can help them set up to do breath analysis with whatever they got. They may not have an Orbitrap. They may not have a GC-GC. They may have just a standard linear quad from Agilent. So we're very unlikely to be able to standardize equipment. Now, sampling and sample handling and the upfront part, we got a better shot at. We are never going to be able to standardize laboratory equipment because you're talking about $500,000 or something or more. But one thing in wearing my editor hat, I see is the really diverse way of reporting data. And Orna touched on this brilliantly. I mean, some people tell you a mean and a standard deviation. Some people tell you a quartile range. Some people actually, go into distribution and geometric mean. I mean, I've been harping on this in papers for 10 years. It is very difficult to compare across laboratory when everybody figures out their own way. Some people do principal components analysis, but never tell you any numbers. I think this is where we can make the biggest impact, is to say, look, whatever you report, at least give me the numbers so that, or at least, well, I like to have my people always give objective numbers, geometric mean, geometric standard deviation, fifth percentile, 50, whatever. It just give me a table at least. Personally, I like to see the numbers because I'm a numbers guy, but I think this is where we can probably help the most in cross-laboratory studies. I'm not saying we shouldn't also standardize everything else, but this is where we got the biggest shot right now.
Yeah, no, I think that's a really good point. It's interesting making a delineation between, so to get to the number, agree with you, different analytical approaches, you can never standardize that. Maybe a better shot on the sampling side, I do think that makes sense. And then, focusing on the reporting. Paul, do you have any kind of thoughts on this topic of, how can we get to apples for apples reporting so you could read one paper and compare to another, see if it's saying the same thing?
Lots and lots. I'm going to have to edit myself. I would say to begin with, I want to commend the Peppermint Initiative to people. If you're new to breath analysis and you want to say, well, how am I doing? Run a Peppermint panel and look at some of the benchmarks that are being published now. And if you're doing significantly better than the benchmark data, you've got a paper, a journal, a patent, something you need to report, because we want to hear about it. I certainly want to hear about it. To give an example, we made a slight change in analytical profile, and we pushed benchmark data from the Peppermint test out by twofold from where the instrument was performing, and then we doubled the time at which we return to baseline values on tracking the Peppermint washout. So it does tell you if you're improving your methodology and you're improving your process, and it absolutely nails it if you're getting it wrong. You know straight away if things aren't right with that. So if you want to reassure yourself that your team are getting it right, or your samplers in clinic are getting it right, then we've found in practice in Sri Lanka, in Canada, in Germany, all over the place, when we ask people to run Peppermint washout tests, we see immediately the team member who's getting problems with the sampling or whatever, and we can go in and support that study and be much more effective and productive in that regard. So that's the first point I'll make. The second point I'll make is this has already been answered for us. All we have to do is go and read other parts of the literature and copy them with pride. And I would recommend the work of Goodacre or Kell or Nicholson and the Metabolomics Initiative and say, well, they've been doing this for blood and urinary and other type biomarkers for quite some time now. Now, breath is different because we don't have pooled samples and things, but we can adapt and adopt. So when Jeremy Nicholson last week was saying, okay, I'm doing 125,000 samples per year through this particular laboratory on blood panels for inflammatory biomarkers and COVID-19 syndrome, then his QC has to be right. He has to have it standardized. He's got 28 separate instruments, and they're all running thousands and thousands of samples a month. To get all of that to align, they have to have nailed the QC, and they've published it, and they've given us their guidelines. So we can certainly start to adopt those in the way that we work and report. And it's something that I'm trying to do myself. And then we can also look at how our clinical friends do it. And okay, so we can adopt consort diagrams, but you can also do consort and workflows for your data processing and data pipelines, and make sure that you describe those accurately. And Goodacre's work, where he took umpteen different pipelines and applied them to the same set of data and got different outcomes each time, shows you that there's as much variability in the pipeline as there is in the sampling and the analysis, for sure.
Yeah, and Roy Goodacre spoke at the conference last year. I think it was a great talk, and I absolutely agree with you. It's, take what works from adjacent fields, and I think that was, an important message from Patrick's talk yesterday as well. So trying to look across not just breath, but biomarkers as a whole. What is some of the kind of best practice and lessons that should be learned that can be applied and ported across to breath? Morad, do you have any kind of thoughts on what, if anything, should be standardized, hardware-wise, reporting-wise?
Yes, I agree with what had been said. When we refer to standardization, is not everyone needs to take the same sample and run it on the same instrument with the same GC temperatures. I don't think it's going to happen anytime, but at least we could standardize and make sure we use good laboratory practice. Okay, the basics, the things that we're mentioning now is not rocket science. Using the right numbers, describing the data, the variability, and so on. But I think, Billy, you asked, how do we make sure that we're looking or comparing apples to apples? So one way is actually to share the data and the numbers, but we need to keep in mind that as long as we're still focusing on tentative IDs, there's a extremely high risk that we're not looking into apples and apples, despite the two papers saying it is an apple. So other than sharing the numbers, looking into the background, making sure it's actually coming from breath, if we keep the risk and low probability of the real identity of those compounds, and we publish those as it's identified, despite it's compared to the NIST library in most of the cases. And yes, in some of those comparisons, we end up with the true identity by comparing it to the NIST library. But I think that we're more moving towards high resolution data. I think the comparison to the NIST starts becoming a bit more fragile. And I think if we take 10 papers that reported the same compound and we go back to their data and chromatograms, we run standards, we'll probably discover that at least 5 of them weren't looking into the same compound. So I think it's a combination between reporting the numbers, looking into the signal to background, and I agree with you, at least the basic data should be as a supplementary material for us to take a look and see what ranges we're talking about. The different weights that Orna mentioned, is it abundance, area under peak, is it TIC, is it EIC, what people report on all kind of stuff that we cannot learn from them, but also the identity of those potential biomarkers. Otherwise, we will not be able to learn from each other as a community. And just a last point, because blood tests were mentioned, I think one of the weaknesses of the data produced currently by the community is it doesn't really allow any meta-analysis. Because we don't know apples and oranges, or if the same apples, and many groups are limited with funds and they end up with small sample size, but that's not a problem if we could compare this data together and perform a meaningful meta-analysis. We could empower those studies together. But for all the reasons that we just mentioned, I don't think it's doable in our field compared to urine or blood tests where the practice is known, the sample collection is known, the analytes are known, and people, they are typically report on absolute quantitation. So you could take 4, 5, 10 projects, combine the data, and potentially discover biomarkers. It's something that we cannot do at the moment, as long as we're not even standardizing the basics of the practice.
Also, feel free to, at any point, to jump in and have follow-up discussions on any points that get raised. A question's come in from Scott. Surely there's strength in having confirmation of results by an orthogonal approach. And I think this relates a little bit back to some of the things that Graham from AstraZeneca touched on yesterday. So often where there's a kind of multi-omic view within studies. So I'm wondering if you may be able to comment a little bit further on undertaking some of these complementary orthogonal approaches as well in the research that we do as a community. Paul, do you want to kick us off on that one?
Yes, absolutely. I think this was a point I was going to make earlier, but I didn't want to be greedy, so I edited it. But yes,
saved it for you for later.
So the genomics, the phenomic profile of what you're sampling is really important data, and we'll do far better, far faster if we start looking at the blood inflammatory panels, for example, or looking at some of the genetic codes and variability alongside the breath data, and how we can combine these different omics, I think is really important, and is the next real chapter in the development of this science. As you know, we've got to standardize, and once we can do that, then we've got a right to actually say, can we play with you, please? Can we start sharing these kind of data, and getting involved in these large multi-center phenomic studies? And the other challenge we have is we've got to speed up our throughput. We really do have to find a way of running faster. Amen, says Billy. But that's,
yes, I hear you.
but that's and I'll put this out there. I think the future of the column is assured for the next 10 years, but after that, I think maybe not. And I think some of the slim devices, the iMobility, three-dimensional stacked drift arrays I've seen, there's some really exciting projects taking place, looking at the role of electric fields and ions, and maybe we'll start to separate things much faster. Before I die, that would be nice, but certainly it's at some point in the future. But yes.
Yeah, I think that would certainly solve the throughput question if we could get mobility separation in, you know, micron millisecond time skills. Morad, do you want to pick it up there, the notion of looking at orthogonal approaches, multi-omic, how we connect back to genomics, etc.?
Yes, definitely. I think I agree. I think we're doing medical studies, medical testing at the end, and it should make sense at some point that through hypothesis-driven testing, we could correlate between the results that we see with other biomarkers in blood or genetic mutations. The phenotype is typically, I don't believe that phenotype could be present only in exhaled breath, because exhaled breath is mirroring something else happening. So at some point after, I think, discovery of potential biomarkers, the biological aspect should be further investigated with combining blood tests, blood markers, or it depends on the application itself, but definitely there should be some sort of a link. It cannot just come from nowhere to be a biomarker for a disease if it's not represented in host response and metabolic pathways that could be measured also differently in the body.
Can I add here, Billy? Sorry.
Yeah, go ahead.
I think it's a very important point. Probably Joachim knows this better than I do, but from my experience with at least the regulatory bodies, they won't accept, I feel like, okay, that they won't accept us telling them like that this is the VOC or this is the biomarker of a disease without knowing the whole process, without correlating to blood or to urine, to some known processes in the body. I think it's mandatory to do this correlation. And maybe, as Morad mentioned before, if we will have the standardized approach for reporting, then we won't need to do everything by ourselves, but we could combine studies together to understand this correlation. But understanding the pathway by correlating to metabolomics, to proteomics, it will be a necessary when we will approach for a regulation. This is my feeling anyway. I hope I'm not totally wrong here.
And Joach, it sounds like, you know, in your work, you would have routinely looked at, you know, liquid phase, urine, blood, and
Yeah.
be interested in your experiences there.
So Orna brings up a really interesting point, and luckily history has resolved it to some extent. When I was first starting to do breath analysis back in like 1991 or so, every time I tried to publish something, the reviewer would say, well, what was in the blood? My colleagues at CDC, Ben Blunt, Don Patterson, well, what was in the blood? And we had to do blood analysis and go back and forth. I think by the time we hit about Y2K, this became less important. Now, as you may imagine, doing volatiles in blood is a lot harder than doing volatiles in breath. And so this question about what's in the blood was always a very difficult one to answer, because storing volatiles in blood was really hard to do. You had to go to -20 and hope like hell it didn't evaporate on you. So we got much better at it, and there was more and more acceptance in the academic community on, you can analyze things in breath and show that they are in breath. One tiny point I want to make is, and some Orna brought up, or maybe it was Morad, about the NIST library. It's generally always wrong, okay? And even easy stuff, like who here has not gotten heptatriene as a hit when it should have been toluene on the NIST library? I mean, this is a simple compound. It's got a 91 base peak, and there's 2 of them that light up almost identically in the NIST library. Once you get to 200 and 300 Daltons, we're running, high res mass spec, and I get 20 different hits for something at 358 with like 4 decimal resolution in Daltons. We need a lot more smarts than just the NIST library to say this compound is this compound. And I think this goes to the orthogonal analysis issue. There are a lot of ways to become relatively sure, but in my last 5 years or so, especially since we started running high res, I'm going, oh my God, everything I thought I was pretty confident about, even running external standards, running through the various computer programs with the chromatographic benchmarks, and you still have a chance of being wrong. I don't know if that's a really big deal, but in LC discovery, about 80% of the things we find, we call features and not compounds. It's got 301.9786. It's got a negative mass defect. We know it probably has some fluorines on it, but sometimes that's as far as you can get. So just a cautionary tale. We've got to, when we publish, make it very clear as to how confident we are that something we call compound X might be something else. And oftentimes it's just an isomer or something like that, but you just never know. And this is, and the blood people have always been super arrogant about this, and they're no righter than we are. I mean,
Yeah, no, that's great. Excellent. So a question's come in from Khaled. So when we're talking earlier about potential standardizing hardware, so I think agreeing that that's pretty tricky. But talking about at least data sharing. So data is getting in the papers, making that freely available to the community as well. And Paul, I know in the Peppermint Consortium, that's a big component of that. So maybe you could pick that up. To what extent should we all be putting our raw data out into the community to help pick over it and dive into it independently.
I've spent the last few weeks curating breath data, and I'm a very small bit operation compared to you and your team, Billy. But looking at how to organize backup and curate 500,000 data files, and each time we do a deconvolution on a set of data, you sort of get another large bundle of files generated and stored and saved, and keeping track of them all. It's a big job. If someone comes to me and says, oh, got to have a look at those 24 sets of data for your COPD panel that you did in 2019 or something, I'll be going, oh, crikey, that's several days work. So there is that aspect that it requires cooperation and sort of funding and attribution at a fairly significant level to make it happen. And there is need to do this. I was looking at a photograph of the moon yesterday, and it was made on a mobile phone, and this individual had produced the most perfect rendered image of the moon at exquisite detail, a more than 200 megapixel image, but it took him 50,000 images and stacked them all in order to get this perfect rendering of the moon. And I thought that's what we should be doing in breath. You know, if I could get that kind of stacking of data, then we would start to get some really definitive resolution there. But to do that is asking for the moon at the moment. Really, it is.
That's very true. I was resisting the urge to shout the pantomime line of "It's behind you." Doc, do you want to pick that one up? You know, is it, you know, maybe there's a desire to share data, but is it actually practical, feasible? Still on mute.
Were you asking me?
Yeah.
Oh, sorry. Anyway, I think 50,000 things is probably out of the realm of reality. In my experience, one of the most powerful tools we have in any of the biomarker research is what we call the interclass correlation coefficient, which is the within and between person variance components. If you take a look at some of the big databases like NHANES, they have a lot of data. They study 5,000 people every 2 years, but they only analyze them once. And so the problem is you get these beautiful distributions, you know, but you don't know how any given sample could vary if they would have done it the next week or the next month. Okay. And you may think, well, that's not important. But if you want to use biomarker data for health effect and compare groups to each other, you really want to know how stable that biomarker is within a person compared to the overall distribution. In the United States, at least, we use the average daily rate of intake of a particular compound to assess the risk of that compound. If it's something like benzene, which could be 10 today, 1 tomorrow, 56 the next day, all depending how close you were standing to your car, it is very difficult to use ADRI from a single sample, right? And so what you want to get a handle on is how does something change between people, or how is it different between people, and how is it different within a person? And I think this addresses Paul's idea of stacking data. And in this case, the stack could just be 2, 3, or 4 samples deep. But I've always encouraged all of my colleagues to do repeat analyses, so to remove autocorrelation. And I think when it comes to encouraging the folks doing research to repeat their analyses, not from the same sample, but take a new sample sometime later, I think is a really valuable tool to tease out the variance components, because they're really important. You know, use cholesterol as an example. If your cholesterol is 200 today, it's probably going to be 200 tomorrow and 200 3 months from now, unless you do something really extraordinary. So with respect to something like cholesterol, which is expected to be fairly stable, it has an interclass correlation coefficient of about 0 or 0.1 or something. You can take a spot measure and assign long-term risk. Take other things that vary a lot, even acetone or isoprene in breath, they change. Run around the track 3 times, your isoprene goes away, right? The within person variance is very important. And so with respect to the whole, soup to nuts of breath analysis, doing things more than once on individuals with some interspersed time is a really powerful tool and can often answer questions that otherwise you're just going, I don't know.
And I think if we're thinking about putting data into the community, I suppose it depends a bit on what type of data we mean. So I get the challenge of raw chromatograms, or trying to ask new things. So chromatograms is very and the tense of both, we're trying to think about how specific compounds vary across time, vary across population. More at the point that you touched on earlier, if we can get to a point where we are reporting on known compounds that are definitively on breath and not just in background, and we can report absolute concentrations of those, we can start to look at how you actually build and integrate data sets across studies and across time that will help unpick some of these questions about how those compounds vary over time. So I was wondering if you could just pick up the theme of data sharing and what type of data we ultimately might want to share in the community.
Yes, definitely. But if I may just comment on what you just mentioned, because I absolutely agree, and I think one of the weaknesses that I currently see in the publications that we see is we don't know to which extent results might be coincidental, and therefore we need to repeat it. And I don't know, Yoke, if you had the chance to be in the presentation earlier this morning about the standardization within the Omni assay, but I described what we at least are doing now and wishing to publish really soon, in which we do what we call the Bob study, breath or blank. We take the same dude in the same room, 10-15 breath samples, 10-15 background blanks, and then we look into acetone, isoprene, and all of those on-breath VOCs and see how they behave in a short period of time from the same volunteer. And the day after, we repeat something similar, but with a population. We take actually 10 volunteers and we look into isoprene or acetone, and we could actually measure for each of those VOCs the intra and inter-individual variability that helped us to optimize the reproducibility of our sampling and measurement, and actually get a feeling about what type of variability, what is the enemy that we are trying to face going forward. So I absolutely agree with your point, and I would love to see the community start sharing this data with different collection analytical methods to see what type of variability and reproducibility we see in general in breath. And to your question, Billy, so yes, going forward, I think as much as I love discovery or untargeted, it has some weaknesses. And I think the biggest weakness is what we're discussing now. It's that lack in the ability to compare data between different projects over time in year 2019 with 2021 because of changes in the analytical method, in the column sensitivity, in the sampling, in the background, and so on and so on. But I think one of the ways that we could overcome some of those problems is by doing targeted discovery. So it's a combination of targeted assay and a biomarker discovery, in which if we identify those on-breath compounds for real with standards that we measure repeatably in a population, we could develop a bespoke targeted method for those compounds and have absolute quantitation against a calculus that is run per sequence. And then we minimize that risk over time. And project A will be compatible to project B, instrument A to instrument C, because we have absolute quantitation. And within the analysis itself, we are actually compensating for changes in linearity, in the limit of detection, in the sensitivity, and so on. And therefore, that allows us to pool data over time, which is almost impossible to do with untargeted discovery at the moment. So it is the vision going forward to have this panel, if you want to call it, like Paul mentioned, panels of inflammatory compounds in blood. We could have panels of on-breath VOCs that we could have absolute quantitation going forward in the future, and combining the data of hundreds and thousands of subjects from different demographics and clinical settings, medical background, and start learning about each of those on-breath VOCs and their behavior distribution among those populations.
Excellent, thank you. I like that you want to compare data from 2019 to 2021. It feels like we all want to forget 2020. Orna, any kind of last thoughts from you on that before we wrap it up for the day?
No, actually, I agree with everyone here, and it's good to hear that we're all aligned. I hope that we will manage to take it a step forward as a community. I like Morad, your final idea of like for each test also performing a quantitative analysis of known VOCs alongside the untargeted analysis. It's a very interesting approach. And yeah, I'm looking forward for the future, for 2022.
Excellent. Well, thank you so much to our whole panel for a really interesting discussion. This is always the one I enjoy the most, so I really appreciate your time. Sadly, yeah, we can't go for a drink afterwards, but yeah, next year conference will be back on back in person. I'm really looking forward to that.